Yes, that
Yeah.
works.
Okay, got it. Okay, so I decided to do something a little bit different. I know I'm supposed to talk about count data, but I wanted to talk about imaging data, 3D imaging and applications to both biomedicine as well as evolutionary modeling. Okay, so I'll start the story with the following. Before Darwin, the classic view of thinking about shapes and organisms was as Platonic ideals. So there's an ideal duck, there's an ideal flamingo, right? And this is what zoologists like Carl Linnaeus and others thought about. And part of the genius of Darwin was that when he started thinking about these things, right, because he was very interested in evolution, he wanted to understand variation. And these are pictures of Darwin's finches. And these beaks have different meanings. And let me just shut my door 1 second. In terms of whether it's easier to eat insects or it's easier to break shells, nuts, right? So the point that I'm trying to make is that variation is fundamental when we think about biomedicine and as well as evolutionary processes. Now, there's another version of this, which was more deterministic and more geometric. And this comes from Darcy Thompson. And if you haven't looked at On Growth and Form, it's a beautiful book. The pictures are extraordinary. And it was really much more from a perspective of developmental biology. And I think one of the things that is very interesting that's happening more and more now is that this interplay between more evolutionary perspectives and variation and developmental biology are interacting with each other more and more. Okay. So one of my colleagues, he studies variation in bones, but we'll talk about other things like that. Sorry, wrong direction. And one of the things they want to do with these are generating phylogenetic trees, but we'll skip that for now. Okay. In terms of shapes, there have been 3 classical models. One is the shape space model by Kendall. Another one is a model based on diffeomorphisms. And the one that we will talk about a lot today is a model based on integral geometry. And I'll explain all of these, but the idea that I want to convey to you is that when you have 3D shapes, there are real, there are representations that are particularly useful for both classical statistical algorithms and machine learning algorithms. So the first example is that this comes from originally Kendall. You go to the British Natural History Museum, you have an artifact, you put down a set of points on it, and you put down a set of points on another artifact, and you know that there is a correspondence between these sets of points. These are called landmarks. And then you basically, what you do is you take the landmarks and you map them to each other, modulo translations, rotations, and scalings. And this gives you a space of shapes. So there's a reasonably dark paper, but somewhat also amusing, about where this was used to try to figure out whether Cromwell's skull is really Cromwell's skull, which is stored at one of the universities in London. The thing that you lose when you use landmarks is nowadays we have these 3D meshes of data, right? These are from micro CT scans. And this happens in both evolutionary contexts, but it also happens when people do micro CT for biomedical applications, such as cancer or other diseases. And if you discretize to a set of landmarks, you lose information, a lot of it. Okay, so then there's a diffeomorphism-based approach, right? Which is where what you do is these are teeth, these are molars, and you take all of the points, you build a little area around them, and then you ask, how much energy does it take to move from one object to another object? And that gives you a notion of distance. And this is the diffeomorphism-based approach. Now, the issue with the diffeomorphism-based approach is the following, is that what happens if your objects are qualitatively different? So these are fruit fly wings, and you have normal ones, and then you have mutants. And the problem is you might have extra veins, you might lose some of the veins, you might get extra areas, right? So how do you model that? How do you model things that are fundamentally qualitatively different? Okay, so the data themselves, right, involve vertices, faces, and edges, right? Because these are mesh data. If you have micro CT scans, and we'll talk about other versions of it, but these are fundamentally the construction. So you can think of this, there's something called an off file, which is a basic data structure that you get, which are the coordinates of the vertices and then the triangles, possibly the edges, but in micro CT, usually don't get edges. And so this, if you want to think about it mathematically, gives you a collection of simplices. So you have points, you have edges, you have faces, and you have tetrahedron, because we don't go beyond 3D. So simplicial complex is a collection of the simplices, with the conditions that all the faces of the simplex are in the complex, and simplices intersect along common faces. So what does that mean? These are things that are not legal. You can't just stick something in between. You can't take an edge and remove one of the vertices, and you can't take a face and remove one of the edges. Okay? So in this setting, we have, again, this is just what I wrote before, simplicial complex. Okay, now I'm giving you something more abstract now, and there's a reason why I'm giving it to you, which we'll come back to later. And this is, actually, give me 1 second. There's something weird going on my screen. Is it okay again? Okay.
Was okay already, I think.
It was okay already? Okay. So there's something very abstract, and this is called an O-minimal structure. And there's a reason why I'm telling you all of this. And the reason is, how many people know how micro CT scans work, right? Or these 3D imaging approaches work?
Raise your
Please.
hands and I'll estimate.
Yeah, raise your hand. And there's no reason you should. I'm just curious.
Nobody knows. Neither
Nobody
way.
knows. Okay, so what you do is you take an object, you put it in a machine, typically made by Siemens, and then you radiate in each direction and you look at the projection. And then what you do after that is from these different directions, you put back the 3D shape. So it's an inversion problem. And this is called the radon transform. Okay. And the reason why I'm telling you this is the math that I'm showing you right here are the minimal conditions under which the radon transform is invertible. Right. So it really relates the physics back to the math and gives you that notion of an inverse problem, right? Of where you can get back the notion of a shape. An example, if your shape, the structure, were a fractal, no hope. Okay. So this relates to something called constructible sets and constructible functions. Okay. I'll make things also more concrete very, very soon. There's a picture of thinking about how shapes evolve and how shapes can be related, which is you have a base manifold. This is your M, right, where evolution is happening. And then what you put on each point in this base manifold is a representation of a shape. And in math, that's called a fiber. And the way you get back from the fiber back to the base manifold is through projection. And these fibers, I'm here showing you examples of monkeys, are really vectors. And they can be thought of, for example, as molars. They can be also thought of examples of tumors as they are evolving. Okay, so what we want to do is model shapes without requiring landmarks or diffeomorphisms. And the way we're going to do this is transform the data into a representation that can be used by deep neural networks or Gaussian processes, right? So basically standard ML/AI or statistical methods. The other thing we want to be able to do, and you'll see why we want to do this a little later in the talk, is be able to pull back the information from this transform space to the original 3D coordinates. There are two summaries I'm going to tell you about. One is called an Euler characteristic. The other one is called persistent homology. And I'm going to talk about this in the shape context. I'm also going to touch on protein structure, but these ideas have also been used in molecular biology context, and I'm happy to discuss that as well. So I'm showing you a picture, and this is Pablo Picasso. And the point of this picture, sometimes abstraction helps us, and it gives us what might be the base structure of what we think about. Okay, so the Euler characteristic for meshes is very simple. It's the number of vertices minus the number of edges plus the number of faces. And so, going from left to right, you see that for these solid objects, it's 2. For the donut, it's 0. And for Swiss cheese, it's -34. So what we're going to do is we're going to take an example. This is a heel bone, and we're going to take a direction, and we're going to ask how the Euler characteristic changes as I sweep through the shape. I'm going to do this from many directions. So for a 2D example, here's a hand. This is what it looks like for the hand. You start filtering, and it changes, and it changes, and it changes. Okay? So what you get at the end of the day is a matrix where each row is the Euler curve in our shape. And what this is actually capturing are the changes in critical points. So maximum, minima, inflection, corners, and discontinuities. And what I have on the far right is what we call the smooth Euler characteristic curve, right? Which is taking the Euler curve and integrating it and zero meaning it. Okay, there's another summary that is sometimes used, and that has to do with homology. So what is homology? 0th degree homology is the number of connected components. So if you have two balls, right? Betty 0 is 2, so there are two connected components. There's no one cycle and there's no void. So one homology, if you look at this, Betty 0 is 1, because there's one connected component. There's one cycle, there's one hole, right? And there's no void. And up to 2D, right? In this case, again, Betty 0 is 1 because there's one connected component. There's no one cycle, but there is a void. I cannot show pictures of higher dimensions. Okay, so an example of this is if you have the curve that you're seeing and you slowly move up, which is called a filtration, and then you look at what's going on the real line below, right? You look at these components and you look at how they come together. And so when we're going to move up a little bit more, right? One of these components are going to merge. And then you keep doing this and you look at when they merge. And so when a new component comes, that's called a birth. And when they merge, that's called a death. And this gives you what's called a persistence diagram. There are other ways of doing this. If you just have point data, you can thicken balls and you can ask how the homology is changing and when it is changing. So formally, a multiset is you have a diagonal which has infinite multiplicity and you have points. So this is what it looks like. I have a red curve here and a blue curve here. And on the right, I'm showing you the persistence diagrams of the two. And the way you can measure distance between persistence diagrams is by asking, what is the minimum cost of moving a red point to a blue point? And because you may not have the same number of points, you can always move something to the diagonal. And this is a Wasserstein metric. Okay. So again, more formally, you can think of the Euler characteristic transform as you take a shape and it's a map from the directions on the sphere to these constructible functions, right? Which are these Euler characteristic functions. So what I have a little bit lower is ECTM. M is my shape, V is a direction, and T is the depth that I've gone to. And again, this is the same thing. It's smooth, so it maps things to functions. And then you have the same thing for the persistent homology transform, where up to dimension d-1, you basically look at the persistent homology in the direction V. Okay, I will skip this. So there's a formal notion of distances between two shapes now, even if these shapes may be topologically different. And I will show you guys real data. Just give me a little bit. Okay, so this is a picture that I wanted to show you. This is a radon transform. So if you collect, let's say a tumor, or let's say you collect a bone, right? And you want to digitize it and put it in your computer, right? The typical way this is done is there's a machine and the machine radiates it and looks at the projection and does this in many, many directions. And from these directions, it gives you a 3D construction, right? And that is how digitization of many, many shapes is done. Okay. There's no need to understand or read this slide, but the point of this slide was that minimal construction, what I told you. There's a theorem by a mathematician named Pierre Shapira, and what Shapira proved is that minimum projection requirements, that's the minimum you need for the radon transform to work. Okay, so what we showed and what others showed is that there's no information lost if you look at the persistent homology transform or the Euler characteristic transform. These are injective maps. You don't lose any information. What this slide says is interesting, which is usually when people do analysis of surfaces and shapes, they have to register them. They have to make sure that they're rotated and aligned in the same way. And what we can show, and we've actually even implemented this in algorithms, and I'll talk about that in a little bit, is that you don't have to necessarily align them. You can think about the directions as random, and each random direction, let's say you get this Euler characteristic curve. And then what you can do is you can use a alignment or an optimal transport algorithm to figure out, how similar they are and to do the alignment. And if someone wants to talk about that, I'm happy to talk about that in detail later. Okay. So my computer, your computer, can't take all directions, right? So the question is, how many directions do you need? So empirically for 2D, we found 162 were okay. For 3D, over 700 directions were okay. But there's a question of, how do I figure out how many directions I need? And first I'll give you a theory version of it, and then I'll give you something we're working on, which is an applied version of it. So this is a sampling theory for shapes, right? What's a complexity metric for families of shapes in terms of the number of directions required to be able to tell apart two shapes in the family? There's a very, very interesting paper by, V.I. Arnold has many interesting papers, but there's one paper where he asks the following question. If I tell you where, for a 1D curve, where the critical points are, can you tell me how many different homological functions are there, right, that have those same critical points? And what's amazing is he gives a closed form solution for this. Okay. The problem when you go beyond 1D is that the ideas that Arnold used really depend on ranking, and beyond 1D ranking is tricky. Okay. So let's say we have a set M in dimension D with delta and K delta, which I'll tell you what they are very soon, as a set of all embedded simplicial complexes in RD. And they have two properties. At every vertex, there's a lower bound on curvature, which I'll tell you what that is. And in any direction, if I look at a little cone, the number of critical points, or how much things are changing, right, in terms of inflections and cusps, is bounded. That's a K delta. So what we mean by curvature, right, is what I do. And let me first say why. Two things that are really, really flat are really hard to tell apart because they look really similar and they're really flat. And so what I'm giving you here and what we're looking at here is a lower bound on flatness, right? This tells me, well, this is a resolution of flatness that I'm willing to resolve. Okay, that's what delta is. Now, there's something that's actually kind of really interesting. So I told you we took this object, right? And in the radon transform, I swept through it, right, using X-rays and looked at the projection. Okay. So what we can tell you is you're sweeping in a direction, right? So you're really asking, how do these directions and how do these Euler characteristic or some other summary change as I go through different directions, okay? And what you can prove mathematically, and it's also true physically, is that what you're really doing is cutting up a sphere. So within each region of the sphere, if I know what that summary looks like in one direction, I can tell you what it looks like in a very similar direction. And basically you're just chopping up the sphere. And the basic question that you're asking, and there's a linear map between these, right? So another way of stating this is the reason why these types of transformations are nice is because when you look at it in the transform space, they're locally linear, right? And having this local linear representation lets you do a lot. It gives you a coordinate system in a way, okay? And so what we were able to prove, and for those with a bit of a theoretical machine learning background, this is interesting because it was using VC theory. We were able to prove how many directions you need to tell apart any elements in this space. And it depends on that flatness, delta, and it depends on the complexity, K delta, and the dimension. Now, I will skip this slide because I don't want to torture you. Okay, am I back? Okay,
Yes.
so let me skip this part, okay? Okay. So what's interesting about this representation, right, is that it lets you start using classical statistics. So there you can use what's called an exponential family, which is a standard model, and you can turn a shape into a matrix. And then you can use matrix models to model the shape. So this is something called the matrix variate normal, right? So you're modeling a matrix. It's an L by T matrix. It has a parameter A that models the mean, a parameter U that models the covariance, and a parameter V that models the covariance between points in a curve. And so you can actually start writing down likelihood models for these shapes. And the power of this is trying to do this straight up with the 3D shape is hard because you don't have a coordinate system. And so now what we've done with this transformation is basically imposed a coordinate system that you can use. And what we'll go look at very, very soon is a non-parametric version of this, how you can use this in Gaussian processes, but we've also used these in neural networks. Okay. So let me just start the data part of this talk, okay? So these are two different heel bones from two different angles. And what we did was we took the persistent homology transform and we measured the distances between the heel bones across 106 primates. And then we did multidimensional scaling to reduce it down to 2D. And what's interesting is at the top right, you see the great apes, and then gibbons are a lesser ape. So spider means spider monkey, baboon, macaque. So then these are giving us some notion of old world monkeys. And then at the bottom, squirrel monkey, howler, sakai. Sakais are extinct. These are new world monkeys. And at the very left, you are seeing lemurs. Now, if you want to make this picture a little bit nicer in 3D, we've done that. You really start getting phylogenetic groups, and you start getting things that we know agree with evolution. So one of the things we also did was we compared this to manually placed landmarks, persistent homology, and automatically placed landmark data. And we also did simulations, but validating this is a little bit tricky, but people thought, well, at least evolutionary anthropologists thought that B looked better. Okay, so I know a lot of you care about medicine. So let's adapt this and bring this back to medicine. And here's a case where we have on the left a brain. We're imaging a brain. It's an MRI. And what you want to do is understand this glioblastoma, right? Which is a pretty severe form of brain cancer. So we have algorithms, we have code that first takes a brain and resects the glioblastoma. Okay, and what we're going to look at now is a case study, an example, where we have 92 patients and they have matched MRI data from the Cancer Genome Atlas. So yeah, and we're going to look at a few features. One are gene expression, where we look at the gene expression of about 9,000 genes. The morphometric features are a set of features that radiologists and oncologists work together to come up with. Some of them are more complicated, some of them are less complicated, but there are about 212 of them. The volumetric features are only 5. They're very simplistic. They're like, how big is a ball? You know, is it more oval or less oval? And we're also going to use these topological features that I told you about. And I'll explain that in a second. Okay, so what are we going to look at? We're going to look at disease-free survival. This is a period after successful treatment where there are no signs or symptoms of the disease. And we're going to look at overall survival. And so that's the entire period after the start of treatment, during which the cancer patient's still alive. And we're going to first ask one simple question, which is, which of these features explain the most variation? Right? So we're using shapes in regression models to predict clinical outcomes. And this type of idea, this instruction, is something that's often called radiogenomics. So what we're really looking at here is an example of radiogenomics. Okay. So for the topological features, we have a bunch of slices of the MRI data. And then what we do is we take a slice and we take a direction and we sweep through it and we look at what the smoothed Euler characteristic transform looks like. And so this is what you're looking at for one slice. And then we do this across all of the slices. So these are stacked 3D and we're sweeping through them. Okay. The model, well, it's going to be something very simple. We're using a Gaussian process model. So the idea that we really have is you have this complicated MRI data or a 3D shape, and then we can turn it into a stacked set of values, which is a curve. And so then you can use any standard tool in functional data analysis, or you can use any standard tool like a deep neural network. So here's a kernel model. On each data point, you put a little bump, right? And you weight these bumps. And so in terms of Gaussian process regression, right, you have a stochastic process. You have the prior for the function. There's a mean and a variance. Typically, we set the mean to 0. And there's a kernel function that defines the covariance structure. And I'll get to that in a second. And what's really, really mostly nice about Gaussian process regression is everything is multivariate normal. And you have closed form formulas for the mean and the variance for the posterior. Okay. Now, what's less nice about Gaussian process regression is that your problem is order n cubed, where n is the number of observations. So we looked at a few kernels. We looked at a linear kernel where you compare U and V. U is what you get from one shape, V is what you get from another shape, and you're basically comparing everything across the direction and how deep you're sweeping. And then we also looked at distance-based kernels. We looked at what's called the Gaussian kernel, and we also looked at something that's heavier-tailed, which is the Cauchy kernel. And so what we did was we did 80-20% splits for cross-validation. And on the left, what you're seeing is disease-free survival. And I'm showing you the root mean square error and its standard error. And the right, what I'm showing you is the percent of times that modality gave us the best results. So what's interesting is these topological features gave us the best results 70% of the time, and they had the lowest root mean squared error. What else is interesting is there's a difference between disease-free survival and overall survival. And we suspect there are good reasons for this. And this is something, again, we can talk about when we get to the discussion. Okay. So this raises a question, which is, well, you can make predictions. Great. But can you explain something? Can you tell us if I had two classes of shape, let's say one is case and one is control, can you tell me what parts of the shape are most relevant in being able to tell apart disease versus non-disease, right? So this is almost like a variable selection problem, but it's a variable selection problem where we don't have any coordinates. Okay, so I'll skip that. Okay, so how do you do this? Okay, so let me first give you a picture and a pipeline, okay? So, and we didn't do this in cancer because we didn't have something controlled enough that we could really tell you whether we were being right or wrong. But what I will say is the method that I'm showing you has been used by people at Genentech for again, case control for psychoactive medication. Okay. So here we're just going to use teeth because we knew more about the teeth, the molars. And let's say you have data from species 1 or some group of species 1, and then you have data from another group of species 1. So again, think of this as your case control. Okay. And then what you do is in each direction you compute an Euler characteristic curve. Okay. And then you find, so these curves give you a notion of a direction and a notion of a depth. And then what you find is in what directions, at what depth, right, are things the most different. Okay. And once you find that, you need an algorithm to invert that, to transform that back into 3D coordinates. And that's the part D. And so this is false color. And what it's telling you is if something's really red, it's varying between the two a lot, case and control. And if it's green, it's not. And what I'm going to talk you through are the steps in this. So we really think of this as a pipeline. And if, for any part of this pipeline, there's something else you'd prefer, go for it, right? You can substitute it. So first, we've already talked about how to represent the shapes, right? So now what we need is a statistical model to classify them. Well, we already talked about that. That's a Gaussian process. So now we need a variable selection algorithm, some notion of evidence of association. And the last thing we need to do is project these back to the original shape. Okay. So the variable selection algorithm we used, and I'm going to talk about this a little bit more in detail in my second talk today, where I'm going to talk about cancer and epistasis for human disease, is a nonlinear model for variable selection. Okay, and I will give you all of the details about it in the second talk today, but I will touch on it now. Okay, so if you have a linear model, how does it work? You do regression, you have x beta plus epsilon, right? And so your beta hat is a projection in a way of your x onto the y. And the standard, least squares or maximum likelihood estimator is based on something called a pseudo inverse. Okay. So that goes back to Gauss. So the spirit of what we're doing is a nonlinear model. You have y is some function f plus epsilon, right? Where f is defined by the kernel model. So what we can do is we can get the analog of an effect size, and we'll call that beta tilde, and that's projecting x back to f. And one way of thinking about this is even in a nonlinear model, locally you're linear. So if you have a representation that captures that, then you can capture that local linear structure. Okay. So in terms of our algorithm, we used Bayesian algorithm, we used MCMC. So we just, step one is nothing but a Gaussian process. Step two is inferring the variance, again, using very standard Bayesian procedures. And then step three is a deterministic step where you linearly project back. So it's reasonably fast. Now, there's a reason why we used a Bayesian algorithm. And I'll tell you about that in a second. Now, the issue, though, is even if you have beta tilde, you can't just pick out the largest ones, because we know that, when you do regression, just straight linear regression, you have correlation between effect sizes, right? Because of the correlation between your coordinates in the system, right? So how do you get rid of that correlation to figure out which are really the most relevant coordinates? Okay, so one idea, and this is called centrality and ranking, is that let's say you have a basketball team, right? And let's ask the question of, if we remove a player, how many extra losses do they have, right? And there was actually a example of this. There's a natural example when Michael Jordan decided to go play baseball. And we can see how much the Bulls lost more games, right? And the idea is players that are more central are the ones for which, if you remove them, right? Your outcome is much worse. Right? So how do you do this formally statistically? So what you do is you look at these beta tildes and you compare your beta tilde, taking out coordinate j, right? Conditioned on beta j. And you compare that to what happens if I just marginalized it out, if I just integrated it out. Okay. And you can think of this in genetics, right? In terms of doing this with variants. And I will touch on that in the second talk today, when I talk about epigenetics. So now what can I do? I can look at the KL divergence between the marginalized version versus the removed version. And the reason why I need to marginalize it is because I need vectors of the same length. And why it was convenient that we had a Bayesian algorithm is because we had multiple draws of it. We could just pretend that it's Gaussian. Okay, so if the scale divergence is 0, then that variant, that coordinate is not needed at all. Okay, so then what we can do is we can write down something which we call rate or relative centrality. And that is, you take the scale divergence of each coordinate and normalize it, right? This just, makes a sum 1. And you can think of, for example, the significant markers or significant coordinates are those for which the rate value is greater than 1 over p. Okay, so that is our evidence of association. So now the goal is, how do you map back? Okay. So the reconstruction algorithm says, pick a cone with a set of directions. For each direction, find all vertices that correspond to the direction and the distance that you're going in, selected by rate. And then you repeat this procedure over all cones. So here's what you can think about. The D features with the highest rate values can be indexed as a direction and a depth. So now for each direction and depth, you need an XYZ coordinate. So you're really missing one value, right? So one way you can think about it is take a small cone and try to solve this linear algebra problem, right? Which is given these directions, V11, V12, V13, that's your first direction. Then V21, V22, V23, it's your second direction. Each direction is coordinated by a triple, because we're in 3 space. And then look at these XYZs, right? Try to solve for these XYZs such that you get these heights. And you do this in an overdetermined system. So this is actually not what we did, because this is a little bit unstable. So what we did was we took these directions, we took these small cones, right? And we just ran the forward algorithm in a way, looked at where they were hitting it in the planes, and then took the intersection. Now, what's really tricky to do in a lot of these problems is how to validate what you're doing is like not insane. So doing the simulations is actually very, very tricky in a controlled way. So what we did was we went to the computer geometry literature, computational geometry literature, and graphics literature, and there are ways of caricaturing. And so what we did was we took Lemurty and we caricatured them, right? And we looked at whether we get back the characters that we made. And these are just ROC curves on this. And yeah, and we looked at a real data set. So a real data set here were, again, lemurs. And what we know are these are their evolutionary history. This is a tarsius. We know that this red lobe got lost as you go further along evolutionarily. And here I'm just showing you how it looks in terms of the false colors, and we're basically getting back what we would expect. So in the last 5 minutes, I'm going to go a little bit fast and tell you we've done the same thing using deep neural nets. We've also done this for protein structure. Slight variation, but again, we can tell you what parts of protein structures are different. We've done this with soft tissue. And the basic idea with soft tissue is that you do a double filtration. You filter over directions and you filter over the value, right? So because these are no longer shapes, right? They're fields, you filter over both. And what I wanted to show you is we actually applied this to a breast cancer data set. And so what you have on the left is a deep neural network that was very, very fine-tuned. And what you have on the right is our, what we call the lifted Euler characteristic transform with just a straight up Gaussian process. And the point is the results are very comparable. Okay, so it's 11, well, my time it's 11:40.
It's 3:10 here. You have about 20 to 25 minutes.
Minutes. So before I get into the evolutionary aspect, are there questions people have?
Any questions? Yes, one over there.
So in the reconstruction algorithm, suppose you want to prove that the directions required are just finite and I don't care about the actual number, does the proof become much simpler?
In the reconstruction algorithm, if you just need to prove that. Short answer, yes. So this is actually a really great question. And this is related to something actually practical. So I gave you an upper I gave you an upper bound, right? But that bound is ridiculous. So what we and others are working on is the following question, and it's related to a project we're doing back at Duke. So glioblastomas are pretty brutal. If you have a glioblastoma, it's probably not going to go well. Now, in terms of brain tumors, there's something called a glioma, which is much more mild. And one thing people are trying more and more to do is for certain cancers, instead of radiating the hell out of people, right, just wait a little bit, give them more mild medication and see if you make sure you don't do something that causes a cancer to evolve in a really bad way. Okay? Okay. So we are doing that for gliomas in a project at Duke, where we are using these representations. First, we segment using deep neural networks. Then we use this representation, and then we use change point models to try to figure out when something is changing, right? And so the patients come in, they get MRI or often CT scans. Now, the reason I'm telling you this is that if you bring people in and do CT scans on them regularly, you're radiating them, right? So there's this question of if a priori I have some collection of, let's say, brain tumors or breast cancer tumor data, right? Can I tell you how many directions I need and which ones they are to get within an epsilon approximation of what I would get from the full CT scan, right? So another way of thinking about it is, can you set up your Siemens machine that it's going to use far, far fewer directions? And the reason why you might want to do that is you're going to radiate the people a lot less, so maybe they can come in more, right? So your question is very relevant to that basic idea. And we're trying to do that by basically thinking of this as a nonlinear dimension reduction problem. And possibly, and we're looking at both variational autoencoders to do this, as well as things a little bit like t-SNE.
Thanks.
Should I go on? Okay. So I'm a Bayesian. So, and I use a lot of Bayesian hierarchical models. So what I'm going to formulate is the following. I have Z is my base manifold. That's where the evolutionary process is happening. And then I have F, which is mathematically, formally, let's say a fiber, but really it's your shape. Okay. And that again is going to be a conditional Gaussian process. And I don't know why I wrote it with the British spelling. Maybe because I'm giving a talk in India. Okay. So on the base manifold, what you can do is you can say it's Brownian motion, or you can say it's an Ornstein-Uhlenbeck process. It's whatever your evolutionary model is that you want to test. And that induces a Gaussian process with a very well-known standard models, which depend on times for recent common ancestors. And because for all of many of these objects, we know, or these organisms, we know the most recent common ancestor, you can do this. Now, I'm going to be talking about this in terms of molarity, but one can do the same thing for cancer evolution. Okay. This slide looks like mathematical gibberish. And so we'll go on. But the point of this slide is that there is a notion of independence. There's an idea here, which is algebraically, when things commute, statistically, you can transform that into conditional independence. Okay, so let's say these are my two molars. I can try to learn a correspondence map between them, right? So a set of diffeomorphisms between X and Y, that's my correspondence map. And then what you can do is, because you have this fiber structure and this conditional independence, you can write down a joint Gaussian process or kernel on the evolutionary model and your fiber. And your fiber is just what is the cost from moving one shape to another shape. Okay. Now, there's something that's a little bit interesting here, which is there's a theorem, there's a result that tells you formally that if the objects, the manifolds, the shapes you're looking at are not isometric to Euclidean space, then when you do this construction of the fiber, right, in terms of these geodesic maps, it's not positive semi-definite. You're going to get some negative eigenvalues, possibly small, possibly big. In our case, when we were working with lemur teeth and other shapes, it was again not positive semi-definite because we got some small negative eigenvalues. Now, if you want to make this go away, what you do is you define a kernel that solves a stochastic partial differential equation based on the Laplace-Beltrami operator and white noise. Okay. Now, what we can do now is you choose two different models. You say, okay, Ornstein-Uhlenbeck, just Brownian motion, meaning there's no selection, right? And let's say our third is something called a burst process. And what we can do is we can marginalize out the fiber, basically marginalizing out the shape. And then what we can do is we can write down a Bayes factor and say which of these models, Bernstein-Uhlenbeck or Brownian motion or the burst process, has more evidence. And the reason why we're doing this is to get a formal model for selection on shapes. And I'm not showing you our results because we're still trying to finish this up and work it out. But yeah. Okay, I will skip this. So there's a lot more to do in terms of this, both in applied and in stochastic work. One of the things we're working on is if I give you a bunch of shapes, if I give you a bunch of brain tumors, right? Can I build a generative model to do this? And one of my collaborators, Lauren Crawford, and his student Emily have made a lot of progress doing this using a combination of neural networks and a representation called alpha shapes. And I think with that, we've got 12 minutes, so I'm going to save some time for us to talk, and I'm happy to give you more details and more explanations.
Okay, Bodha?
So I have a question which is
Yeah.
from the earlier slide,
Uh-huh.
which is regarding rate, which is relative centrality. So what I saw
Uh-huh.
was that you wanted to see how, like, kind of like which feature is more important, and you trying to remove the correlated feature.
That's exactly correct.
Yeah. So what I'm, but in one scenario, like mostly in medical data, lots of it's multidimensional. So there are multiple features which is available. Say, for example, two features are highly correlated. Let's call it feature 1 and feature 2. Feature 1
Yes.
is feature 1 comes like the important feature, but and then you tend to remove feature 2 because feature 1 is kind of representative of feature 2. But say, for example, feature 2 and feature 3 complement each other to predict better than feature 1. Then is this an optimal way to find a optimal space of feature, or something else should be applied?
Okay. Yeah, this is a really good question. So this will be related to actually my talk later today. And the point here is that we cannot prove that this is optimal, number one, okay? Number two is if you look at what we're doing, we're only looking at single removals, right? Now, what you are suggesting is what happens if we looked at all double removals, all triple removals, right? And in theory, you can do that, but it's going to be computationally brutal. Okay? And so I'm actually going to talk about epistasis in my second talk today. And there we will be talking about things that look more like what you're suggesting, using an extra marginalization step. Right? Because here you're only removing one at a time. It's not a greedy search, right? Because we're just looking at everything as a multivariate normal, right? And just measuring each one, right? But it is only removing one. It's not doing all pair removals or all triplet removals or all so on and so forth. Does that answer your question?
Yeah.
Okay, Tav? Yeah.
Thank you, Sayan. So I just noticed that the accuracy gain that you got with the topology-based features was actually quite small, from maybe 0.6 to 0.62, from 0.6 to 0.62, if I remember correctly. do you think, so what's a computational, requirement for computing these features, and how robust are these two outliers? Is my first question.
This is what you meant. This is a picture, right? Yeah. So what was it? Yeah, 0.942 to 0.803.
So the next one that I'm seeing is so so I think 0.958, and then the previous one is 0.964. Am I looking at
Oh,
the
yeah,
right?
yeah. So this is, okay, thank you for bringing this up, okay? So in overall survival, you don't do much better. You really don't do much better. And I think there are a few reasons for this. So there's certain things we know, which is for example, in a breast cancer, if you have a void, right, you have a lot of necrotic tissue, so you're going to do better. We also know in brain cancers and in other cancers, right, if you have more, if the set of connected components is larger, the outcome's going to get worse, right? And again, this is very much part of the disease-free survival part of this, right? So I think you see a reasonable difference in disease-free survival, but as you observe, you see a lot less difference in overall survival. In terms of the computation, the volume one is the fastest because you only have 5 features. The gene expression and the SCCT, once you computed the features, are basically equally fast. The morphometrics is a little bit faster because you have an order of magnitude fewer features. In terms of computing the features, the morphometrics is the most costly to compute. Now, what I'll tell you is if we had methylation data, it would beat all of these. But there's a point here, which is when you're looking at the brain, right, you can't, getting gene expression data and things like that, or methylation data is complicated, right? It's very, very, very invasive, right? So if you have an imaging modality, that helps you a lot.
Thank you. And I'm assuming that the numbers in braces are the confidence intervals?
Yeah.
Okay. And are these methods sensitive to outliers?
I don't know. I mean, my guesses are probably all sensitive to outliers. I think
because the way you were describing the construction of the filtration diagrams and the persistent homology, it just occurred to me, what if there was an outlier point, and how much would that affect the downstream features?
Yeah. So in a way, probably. Okay, let me restate what I'm saying. I think the reason why the Cauchy kernel worked the best, and that's what I'm showing you the results for, is because of that, because of the heavy tail. So I think if you use a heavy tail kernel in a Gaussian process, you will be more robust to the outliers. I think the features are not crazy sensitive. I think also things like some of the, like a deep neural network, if you had enough observations, would also be robust. If you don't have enough observations, it's not. So I think, things we find is, because in some of the protein structure examples, we had many thousands, right? And the deep neural network did great. But in the cancer case, because we only had about 100-ish, right? The Gaussian process did better.
Thanks, Sayan. Very interesting work.
Yeah.
Thank you, sir. So I want to confirm what I understood from your presentation.
Mhm.
So you are, so you can tell me whether I'm understanding it correctly or not. So you are proposing a method which is statistically and mathematically very sound, and that is for classifying the image based image based yes no kind of categories, right?
Yep.
And from that image you are trying to extract features by using the concept of geometry, and on those features you are trying to uh, uh, use some non-linear transformation that you have you have talked about earlier, like smooth, by using some smoothing non-linear transformation. And based on those transformations you are trying to predict this survival thing.
That's exactly correct. Now, there are two versions of this we want to extend to, and that's really tricky. And I'll tell you why. So one version is if instead of 0, 1, right, you're trying to predict a real number, right? Like, high blood, amount of high blood pressure, right? Or something that's more continuous, right? One example. The other example is when your data, the individuals are not all IID, right? There's some relatedness structure, right? Can you think about some type of mixed model? And what I can tell you is we can come up with an algorithm for both of these. The problems that we've had is how to do a reasonable simulation to, have faith that what we're coming up with is actually sensible. So one of the real challenges that we have been facing in this type of radiogenomic shape data is how you do a reasonable simulation, that you have some idea of how you're controlling.
Okay. And your algorithm is applicable for both dichotomous as well as the count outcome, and or maybe a more continuous outcome.
We can adapt our algorithm to count data. We can adapt it to continuous outcomes. I know it's totally doable. It's just that we know how to do it. We just haven't done it because we haven't yet figured out how to do a clean simulation that we will understand.
And somewhere you have mentioned about the matrix normal distribution or something. So I want to understand what is matrix variate distribution, or can you, yeah.
Okay. You have a matrix?
Yeah, this one, yes.
You have a matrix, okay?
So is it a multivariate normal distribution, or means how it is different from the?
This is a great question. So it's related to the Wishart distribution. It's a generalization of the multivariate normal. So if you think about a matrix, right? You have two directions, rows and columns.
Yes.
Right? So like any other distribution, you need to specify a mean or infer a mean, right?
Yes.
That's A. That's fine, right? But because it's a matrix, right, the simplest construction you can think of is that there's a covariance across the rows and there's covariance across the columns.
Okay.
And that's what this is modeling.
Okay. So
Right? That's why you have two parameters. One is a covariance across the rows. Let's call that U. And V is a covariance across the columns.
Okay. Thank you, sir. So yeah, so I will go back and read these models again. Thank you.
Yeah, so it's called the matrix variate normal. It's related to what's also called the Wishart distribution, right? It's a slight generalization of the Wishart. But there's a book by, I think, Anderson on various types of matrix distributions that will tell you about this in very, very painful detail.
Okay, sir. Thank you.
Okay, any further questions? Okay, so let's thank Sayan for this very interesting talk, and